Myself, Dr. DR.MANOHAR, Assistant Professor of Chemistry from Government Degree College, Siddipet, Autonomous. My topic is analysis of nutrients in soil samples in and around Siddipet. These are my co-authors. Abstract. Soil analysis can provide important information about physical conditions, status, and chemical properties that affect a soil potentiality growing plants. In this paper, the soil samples were collected from villages in and around Siddipet district, Telangana, and studied for the estimation of macroelements and microelements like nitrogen, phosphorus, potassium, manganese, and copper. Slightly below the values of these nutrients are below the reference values. Uh, The analysis is targeted to provide the necessary information required to set the target of nutrients application. It also monitors the changes in the parameters of soils and thereby help to balance the nutrient application. Purpose of project: deficiency of nutrients like nitrogen, phosphorus, potassium, and other micronutrients is leading to yellowing of leaves, leading to less yield, excess labor, expense, ultimately loss to the farmers. Our motto includes identify such nutrients deficiencies and recommend the proper dosage to farmers. Judicious use of fertilizers based on soil test results. Reduction in indiscriminate use of fertilizers resulting in reduced ill effect on soil. Objectives: to identify and collect the different soil samples in a random Siddipet, to analyze macroelements and microelements, to estimate the nutrients. Introduction. The soil is the most abundant and precious resource in the earth that needs to the managed carefully, contains many essential nutrients, organic matter, and many useful microorganisms. India has many types of soil such as alluvial soil, desert soil, mountain soil, red soil, forest soil, etc. The alluvial soil is the most available, which covers 43% of earth soil, and follows red soil 18.5% and black soil 15%. And Telangana has red gravel soils majorly, also various other types like chakas, red sandy soils, dubbas, deep red loamy soils, and very deep black cotton soils. In this newly formed state, the study of soils has great scope. In that too, Siddipet is the most recently formed district which attracts our attention. So, with the purpose to study the self-sufficiency of our soil, we consider 10 samples near areas like Chalmeda, Podgal, Bumpalli, Siddipet, Kondapak, Chinakodu, Nangnur, Rajagopalpet, Asamirapur, and Apanpalli. This leads to the identification of deficient elements, mainly micronutrients, macronutrients like nitrogen, and can add to overcome to the deficiency such element. The soil fertility and its evolution is the area which needs immediate attention, as an arrest in the productivity of several crops is due to the ever decreasing soil fertility and imbalanced application of plant nutrients. Potassium assumes the greater significance among the essential plant nutrients because it crop production produce and improves and nitrogen and phosphorus use efficiency. Research methodology. First, collection of soil sample. We have to collect the sample from an area exceeding 0.5 hectare, can be treated as a single samples unit. Fertilized plots, channels, areas near trees, wells, composites, files must be avoided for the sampling. Depth of sampling is an important consideration for collection for cereals, vegetables. The samples drawn from 0 to 15 centimeters. The soil sample is collected by quartering process, in which the entire soil mass is divided into 4 quarters. Discard two opposite ones and remix the remaining two. Repeat this process until about 500 grams soil is left. Analysis of macronutrients. Soil and water should be taken in the ratio 1 is to 2.5 and kept in a shaker machine for 5 minutes to prepare soil solution or soil sample. By this soil sample, we have to calculate pH, EC, OC, nitrogen, phosphorus, and potassium. You see pH, the above solution keep in pH meter and calculate the pH of the solution. Then EC, above solution keep in conductivity meter and you calculate conductivity of the solution. OC, for OC you have to take different solution. So 1 gram soil and 10 ml 1 in potassium dichromate solution taken in 500 ml conical flask and add 20 ml concentrated H2SO4, then add 200 ml water, then add 3 to 4 drops of orthophenanthrene indicator and titrate against 0.5 N ferrous ammonium sulfate. At end point the solution turn into blue to red. The readings are noted down and calculated the OC of the solution. Then nitrogen. For nitrogen you have to take 5 grams oil and add 25 ml potassium permanganate, add 25 ml boric acid, then keep in the solution at calculus machine up to 9 minutes. Then we observe the color of the solution is in green color, then titrate against 0.1 HCl. At end point solution color change to red. Then readings substitute in equation and calculate the nitrogen percentage in that soil. Then phosphorus, 5 gram solution, add 100 ml water and add 40 ml sodium bicarbonate, keep in shaker for 30 minutes. Then color absorption by adding charcoal. Then you have to take 5 ml soil solution, add 25 ml boric acid in UV visible spectrophotometer, keep in this machine and calculate the phosphorus by suitable equation. Then potassium, 5 gram soil, add 25 ml ammonium acetate in conical flask, keep in shaker 5 minutes. Then after we have to filter with Wattman number 44. Then the solution keep in flame photophotometer and note down the readings in the suitable equation and find out the potassium. Next one, macronutrients. The soil solution pH below the 6.5 you have to take first procedure and above the 6.5 you have to follow second position. In that first position, shake 2 grams of soil with 20 ml of 0.1 HCl for 5 minutes, filter through watt number 42, filter paper, collect the filtrate and estimate using the atomic absorption spectrophotometer. In the second method, shake 10 gram of soil with 20 ml of DTPA, diethylene triamine pentacetate for 2 hours, filter through Wattman number 42, filter paper, collect the filtrate and estimate the contents of iron, manganese, zinc and copper using the atomic absorption spectrophotometer. You have to note down the digital value of these nutrients. Then these are the results. So based on these results, experiment results, you have to find out that some of the samples are above the reference values, some of the below the reference values. Here the given S1, S2, these are the samples from different villages. Reference means district average values. Then ISSS, it is a Indian Society of Soil Sciences reference values. So you have to compare these values with reference values and ISSS. Based on these results, we have to find out this one. These are the graphs which are indicating the which samples are below the reference values, which samples are above the reference values. If you deficient in the some nutrients, what will do? Next one, we have to give a conclusion and suggestion. So these are the conclusion and suggestions. Finally, we notice that most of the test values are lower than the normal ranges. So we need to add fertilizer to the soil to improve soil fertility. All the samples were found nitrogen deficient. So farmers are advised to go for the application of nitrogen fertilizers Government recommended for split application of nitrogen fertilizers 3 to 4 times at basal, vegetative and harvesting stages. Fertilizers like ammonium phosphate, calcium ammonium nitrate, urea can be supplied when irrigating. Additional water ban crop needs can be used to flush excessive salts below the root zone and maintain EC level, which is based on crop tolerance. Good soil management measures to maintain soil organic matter and overall soil health must be utilized to maintain the desired EC level. Sample 4 and 6 are having more EC value than reference, so need more care. Broadly, soil was sufficient in phosphorus with the least value in sample 3 7. So DAP is recommended. Them sample 7 was good in potassium level. So need to focus on nitrogen and phosphorus separately. Even the soil was found with sufficient potassium levels. So normal recommended potash quantities are applicable. All the soils are rich in micronutrients like iron, zinc, manganese, and copper. Slightly iron deficient sample like 5 need an inexpensive and commonly used material for the purpose is ferrous sulfate. Sample 8 was found most richest lowest in manganese. Manganese sulfate can be supplied with copper sulfate to overcome slight deficiency of copper. These are the recommendations and conclusion of the paper. So, thank you. Thank you, everyone.